Let's quickly over the sliding filament model and the major filaments, myofilaments. So the thick filament is myosin, that's in blue. The thin filament is actin. So when the size of the band changes, so when the muscle contracts, the Z lines, so these lines at the end of the sarcomeres are Z lines. So 1, 2, 3, 4 Z lines are currently visible. So when the muscle contracts, the Z line is brought closer, both Z lines are brought closer to the M line, which is the line in the middle. So note the change in the size of band. So the Z lines are brought closer to the M line. The size of the A band remains the same, and the length of thick and thin filaments does not change. They just slide over each other. The only thing that changed is the I region over here, so the I band. Okay, but keep in mind the length of the filament remains the same. During muscle contraction, there's something called as a cross bridge formation. Now, when the myosin head binds to the actin, so this is myosin over here because it has the globular heads, and this is the actin. The two most important components for this to happen is calcium ions and the attachment of ATP to the myosin heads for energy. Okay, so the myosin head binds to the active site over here of the actin filaments and pull the actin. So let's look again. Calcium binds, tropomyosin releases the active site, and the active site is exposed. The myosin head binds to the active site and pulls on the actin to cause contraction. Now, this formation between the actin and myosin is called the cross bridge formation. Okay, and the cross bridge cycling itself refers to the repeated attachment of myosin and actin to cause contraction and relaxation, and again contraction and relaxation, and so on. Okay, so all types of muscle cells contract in this manner. Okay, so just understand the cross bridge formation overall before we delve into the details. Okay, and two most important components are ATP and calcium ions. So ATP releases energy when it breaks down into ADP and PI. ATP is adenosine triphosphate, ADP is adenosine diphosphate. And when the bond between the last phosphate is broken, ADP is formed, and the breakage of the bond releases energy along with phosphate. So you don't need to know this reaction for the exam, but you need to understand this to understand the cross-bridge cycling. Okay, so let's start with the first step. So this first step is basically the step you're seeing is left from the previous cycle. The first step says myosin head attaches to the actin myofilament and forming a cross bridge. So in this step, myosin is loaded with potential energy and it is attached to the active site on the actin filament from the previous cycle. Okay, so at this point, the myosin head is loaded with energy and it's ready to pull on the actin, but it hasn't pulled on the actin yet. And this is called the high energy configuration. Okay, so the next step is where the actual work of pulling happens. Okay, so let's go back to the first step. In the first step, ATP has broken down to ADP and phosphate, but they are still attached to the myosin head. So ADP and PI are still attached. So as the myofilaments move into the next step of the cross-bridge cycle, ADP and phosphate are released, and this release causes a change in the conformation of the myosin head. So the myosin head pivots, and as it pivots, it pulls the thin filament towards the middle line of the sarcomere, and this is called a working stroke. Okay. Now, after the myosin changes shape, ATP, another molecule of ATP, binds to the myosin head, and this causes the release of the myosin head from the actin filament. Okay, so this is called the detached phase. Okay, because the myosin head detaches from the actin when ATP binds to the myosin. And then the fourth stage is again hydrolysis of ATP. When ATP hydrolyzes, it breaks down into ADP and PI. And the ADP and PI still remain attached to the myosin head. And this breakdown of ATP causes a change in conformation of the myosin head again. Okay, so the myosin head gets cocked in position, so it stretches out to attach to actin yet again. Okay, and the myosin head again attached to the exposed active sites. And the cycle repeats. Okay, so this link over here shows a nice animation for the cross-bridge cycle. Please don't forget to look at this animation because I think the animation provides a better idea of what's happening in the cross-bridge cycle.